Hello friends. Hello friends. Today we are going to see the new topic, that is the starter culture. Starter culture is very important. But before before giving the definition of the starter culture, I would like to give some introductory part of the starter culture. What is starter, and why this topic is added into the syllabus? This is the important question. Starter culture is nothing but it is the selected strains of microorganisms which is used in fermentation process. In Marathi language, when uh, just in a single sentence, the definitions or the meaning of the starter culture is uh, virzan. Virzan means starter culture. Starter culture means the virzan. And why this starter culture is used? This starter culture is used in the fermentation process. It is used in the preparation of the curd, in the preparation of the fermented milk product. So it is the selected strains of the microorganisms which are very useful. Uh, this is the introductory part. As I told you earlier, this is the starter cultures are the selected strains of the microorganisms, and they are used in the preparation of the products like the dahi, yogurt, cheese, and these are the selected or desirable active microorganisms. Uh, and these microorganisms deliberately added to the milk to convert the milk into dahi, and these microorganisms converts the lactose into the lactic acid, and due to the addition of these microorganisms the important changes occurs. For example, due to these desirable microorganisms, due to these starter cultures, specific changes like the appearance in the body, whereas the milk is the liquid and dahi is the uh, semi-solid. It The dahi, milk and dahi is again has a different body texture, whereas the milk and dahi is again also having the different flavor characteristic. So these changes is occurs due to the starter culture. Again, some important points regarding the starter cultures, regarding the introduction to the starter culture. Due to the starter culture, more recently, it is said that many researchers are also working on the benefit of the starter cultures, and in their research, it is found that the starter cultures are also very useful in suppressing the cancer cell, in suppressing the cholesterol level, and they are also very useful in digestion. So this is all about the starter culture. This is the some introductory part about the starter culture. Now let's see the advantages of the starter culture. As I told you earlier in the introductory part, these starters are also having the more advantages, and therefore they are deliberately added into the milk. Due to their advantages, they are deliberately added to the milk for the fermentation process. The number one advantage of this starter culture is uh, they enriches the human diet. And how they enriches the human diet? They enriches the human diet uh, for the various flavors, for the aroma, and for the texture of the food. So this is the number one advantage, is that it helps in the enrichment of the human diet. Also, it helps in the preservation of the food. by producing the different acids like lactic acid, alcohol, acetic acids, and alkaline fermentation. By this way, that helps in the preservation of the food. Again, these starter cultures are very useful, or they helps in the bio-enrichment of the food in the way they provide the proteins, essential amino acids, essential fatty acids, and the various vitamins. Starter cultures also helps in the digestion. The person or the people who suffers in digestions, for those people, the starter cultures or dahi or the fermented foods play a very important role. Also, they helps in the promotion of the growth. Again, these starter cultures kills the harmful bacteria present in the gastrointestinal tract. And I think due to all these benefit, the dahi or charge or tab, or you may say buttermilk, is a inseparable part of our Indian diet. Due to this these advantages, the many people of India generally have a charge or tab or buttermilk uh, during the meal. During meal or midday meal. Again, uh, starter culture also helps to suppress the cancer cell, suppress the cholesterol or tumors. So these are the few important advantages of this starter culture. Again, one more important of the starter culture is that the persons who cannot digest the milk, those persons are called as the lactose intolerant people. For those people, the starter cultures play very important role, as they cannot convert the lactose into the lactic acid. For those people, the starter cultures convert the lactose into the lactic acids, and becomes their way very easy for the digestion of the lactose. So these are the few advantages of the starter culture. Uh, I think in the introductory part and in the advantages part, we have seen a lot of things about these starter cultures, and by this way, you might have got the definition of the starter culture. But once again, I would like to explain the definition of the starter cultures. Starter cultures, here in these slides, three different definitions are given. Number one definition is that the starter cultures are the bacterial or the fungal strains, either pure or mixed, used to initiate a fermentation process. It means that a desirable starter cultures or desirable strains or desirable fungal strains are used in combinations, and those are used in the fermentation process. Those are used in initiation of the fermentation process. So this is the first definition of these starter cultures. The second definition is the again means the selected strains of the food grade microorganisms, means the microorganisms which are beneficial, the desirable for the human being. Only those microorganisms are selected in the laboratory, and those microorganisms are used in industrial use for the fermentation process. And due to the fermentation process, a desirable appearance, body, textures, and the flavor is produced by these selected microorganisms. So this is the second definition, whereas the third definition is the starter culture means the microorganisms that are selected based on the ability to produce the lactic acid, the curd productions, low pH, and prevent the spoilage, produce metabolites that gives the desirable flavors, or produce the enzymes that ripen the dairy products. It means just we have to keep in mind that the microorganisms are not, the starter cultures are nothing but these are the selected strains of the micro or organisms that are deliberately added to the food for the fermentation process, and those are called as the starter culture. Here a few functions are enlisted. The different functions like the acid production, flavor, preservation, gas formation, stabilizer formation, the lactose utilizations, lowering the redox potential, proteolysis and lipolysis, miscellaneous compounds, and so these are the few functions of the starter cultures. In the acid production, what happens? Due to the acid production, what happens? That is very important one. Due to the acid productions, uh, it helps in expulsion of whey during the butter manufacture. It also helps when the acid is produced in fermentation process, when the selected strains of bacteria is added, microorganisms is added into the milk, so that lactose is converted into the lactic acid, and due to this conversion, it helps to milk for the preservation, means to increase, it increases the shelf life of the milk. Also, it helps in the development of flavor. A particular type of flavor is produced in dahi. Then, due to the starter culture, a particular type of flavor is produced, and that flavor is produced due to the diacetyl or acetaldehyde, and this flavor is produced by the starter culture. Preservation. Uh, Starter culture also helps in the preservation of the milk and the foods by lowering the pH and redox potentials. As I told you earlier, they are also produced. They converts the lactose into the lactic acid, so the lactic acid is also produced. Also, they produces the antibiotics. They produces the H2O2, and again they produces the acetate, and so all these compounds helps in the preservation of foods. Some Italian cheeses, the characteristics of the some Italian cheeses is the formation, eye formation, and these eye is generally formed due to the gas formation by the starter culture. So the eye formations is the main characteristics of some Italian cheeses. Then, due to the gas formations, a texture or blue vein production, uh, the production of open textures, for example, in blue vein cheeses, is also developed due to the starter culture. Then, the stabilizer formation, development of body and viscosity. For example, the polysaccharide materials. These polysaccharide materials are also formed due to the starter culture. Again, as I told you earlier, the persons who suffers the digestion, the person who cannot digest the milk easily, for those people are called as the lactose intolerant people. For those people, the starter cultures or fermented milk products or the foods are the best choice. So, the starter cultures also utilizes the lactose, and they convert the lactose into the lactic acids, and for the persons, the lactose intolerant people, this is the very useful. Then again, by lowering the redox productions, it helps in the preservation of the food materials, helps in the development of the flavor. Then proteolysis and lipolysis, helpful in ripening or the maturation of cheeses. Generally, cheeses are ripened uh, for a month to produce a desirable flavor, to produce a desirable textures. So the starter cultures helps in the proteolysis and lipolysis in the cheeses, and the miscellaneous compounds means the starter culture also helps in the production of alcohol in milk products such as the kefir and kumis. So this is all about the functions of the starter culture, and in our second part we will see the